It was a sad occasion when we lost Andy. But today is not a sad day. Today is a happy day, because today we're here to honor Andy Blei. We're here to remember his humor, his integrity, his help, as Rajiv alluded to. Andy was a terrific scientist, wonderful doctor in all senses of the word. He was a gentleman. Are there many other gentlemen in biomedical research? I sometimes wonder, and I include myself in that. Andy was indeed a gentleman, and he was my friend, and he was a good friend. The picture that speaks 1,000 words is on the screen. We jogged, we walked, we talked science, we talked history. We had a common interest in modern history, politics, football. We had many common interests, as he had with so many people in this society. And I'm very grateful to the society and to the council and to the committee who honoured me by awarding me the lectureship today that I'm going to speak to you about. We met all over the place. This was in Dusseldorf. It was not an ISHEN meeting, I believe. It was another meeting in 2004. Or was it the ISHEN meeting? No, I think not. This is Andy and I in a meeting in Morioka. Andy was a bit of a rebel. You can see where his hands are. He'd been told about 6 times that his hands had to be on his knees. And we kept retaking the shot because Andy didn't like to have his hands on his knees. So, yeah, he was forever the rebel. And what we're going to do in about half an hour from now, we already did in Val David. And you might want to take a copy of this, those who are interested. You'd be interested probably in trying to identify some of the people. There's Chris Rose here in a former life, front and center. Myself, Andy. Mario Baraldi, next to the left-hand end there. Mario Baraldi, I wonder what happened to Mario. Sol Colchon, so many old stalwarts. You'll notice there are not an awful lot of females in this team, and that has to change, and I believe it's going to change today. But yeah, great, great memories. I could go on and on and on. I won't, because we are tight for time. And I thought I would. Andy was a great one, as we all remember, in re-looking at things that had gone in the past, or in putting things together, synthesizing ideas, and you would get that kind of sparkle in his eyes and say to you, Rajiv, we have to do this, you know, we got to move on this. So I was looking through some of his most recent stuff, and I came across this editorial he did on basically based on an article by Debbie and Rajiv on synergism between inflammation and ammonia in hepatic encephalopathy. And I thought, Andy had often talked about synergism, and you know, I've heard it mentioned many, many times at this meeting. I thought it might be an interesting vehicle to just talk a little bit about where we are with it and how much Andy played a pivotal role, as he did in so much, in hypothermia, in synergism, in so much. I started out by looking for the definition. Get into the science now. Synergism is the interaction of discrete agencies, agents, or conditions such that the total effect is greater than the sum of the individual effects. And I have to say that the synergism I've been listening to, including my own, from my own articles, I don't think they fit the definition. I don't think we've always shown that the effect, total effect, is greater than the sum of the individual effects. You can have a look at that as we go through a few examples and let me know what you think. It's not here I'm not here to try to criticize what we've done and what we've said. It's just a humorous way of maybe getting through the issue of synergism. But there is a clinical, potential clinical importance here. And I come back all the time to reminding myself, I even have to remind myself that hepatic encephalopathy is a neurological disorder. And it differs from all the other neurological disorders that I've been involved in, Alzheimer's, Parkinson's, epilepsy, stroke. It differs in that we think it's caused by one thing. And so our clinical management is based upon treating one thing, lowering ammonia. Maybe a little bit of infection involved, but that's about it. Every other disease I know that's a neurological or even a psychiatric disease, patients are treated with a cocktail. 8, 10, 12, 15, as many as you can find, because we found all kinds of mechanisms involved in the cognitive dysfunction in these disorders. And I think we could and should perhaps move in the direction of looking at some of these other factors. We've started, I think, with the inflammation, ammonia story that I alluded to. That's a few light-hearted things first. Where did it start in hepatic encephalopathy? Well, it started with Les Zeev, super guy from Minneapolis. The agents that are shown in white here were the agents that he looked at. Les had the idea, he was a wonderful person, he always brought his wife, and she was obsessed with an interest in pied wagtails, kind of little bird, and she would come and sit out in the Kevin's laughing because he can remember it. Sit out on the lawn while we were talking science, and she would feed the wagtails. Les was passionate about synergism. He thought that ammonia combined with mercaptans, combined with phenols, combined with octanoic acid, and this mixture was what caused hepatic encephalopathy. He wasn't wrong. He particularly was interested in methane thiol. And it's obtained in crystalline form from fulminant hepatic failure patients' urine. He found it in small amounts in reversible coma in animals. Serial blood levels correlated with HE severity. It acted synergistically, he said, with ammonia. And neomycin reduced its production in the colon. Now, if somebody came up with that today, we'd all be jumping up and down and thinking, we've got to look at that. Andy would have been saying, Rajiv, we've got to look at this. Here was, here's the basis for Les talking about ammonia and methane thiol. This is a smelly, horrible liquid, probably gaseous compound at room temperature. And he showed that methane thiol, when present, lowered the dose of ammonia that was required to induce coma. And that was the basis for him talking about synergism. And he did many studies like this with the other compounds too, phenol, octanoic acid. Do we want to relook at this? I think if I was coming into the field, I might want to relook at some of this. Interesting. I've just picked examples here. They're not picked for any particular reason. I probably already had slides for some of them. That's probably why. Let's talk about manganese. Normally removed by a hepatobiliary route, increased in brain in chronic liver failure, causes T1-weighted hyperintensities in globus pallidus and in other areas of the basal ganglia on MRI. And the signal hyperintensities are correlated with motor symptoms. Here are the images in the lower. We don't seem to have a pointer, do we? Or is it working now? Oh, it is. Hyperintensities here in globus pallidus, as I mentioned. We microdissected these from our brain bank in Montreal many years ago, measured manganese. You can see here massive increases in the patient material. And this Later on, we exported one of our best fellows, Laurent Spar, to Geneva, and the folks in Geneva are still having a great time with him. Laurent went there and convinced the neurologists, and there's quite a feat, convincing neurologists to get interested in hepatic encephalopathy. We always had two. We had Karen and we had Alan Lockwood. And Alan isn't so active now, and Karen is our resident expert neurologist, the only one I think who's really ever shown much interest in hepatic encephalopathy for any period of time anyway. Anyway, he managed to convince them that it was important, and he's carried on this work in Geneva with them. Such that they, using better equipment than we had in Montreal, they showed that these hyperintensities were also present in substantia nigra. And substantia nigra, if there's manganese deposited there, and if it's caused damage, then you're going to get a Parkinsonian type syndrome. And in fact, that's what they found. And this gave birth to this really, I think, is becoming a new entity, cirrhosis-related Parkinsonism. Quite prevalent, somewhere between 5 and 25%. The numbers are very unsure because so few people have done measurements, but it could be quite high. They found 22% in patients in the transplant wait list in Geneva. What about chronic liver failure? Let's have a look. Ammonia and manganese. Both cause, how could they possibly be synergistic? We know that ammonia is present throughout the brain. We know that manganese is present in basal ganglia. Both cause decreased uptake of glutamate by the astrocyte. Let me just remind you now about the glutamate system. Here's the presynaptic neuron, postsynaptic receptor, astrocyte that picks up glutamate that having been released from the presynaptic terminal using this transporter here, EAAT2. Remember, there's this glutamate-glutamine cycle, molecule of ammonia taken up here into glutamine, shipped back and released here. So ammonia removed by the astrocyte and produced in the neuron. Here's the data from Alan Hazel in our lab in Montreal, and you can see that ammonia here causes a decrease in expression of this transporter. Manganese alone causes a decrease in this transporter, and the two together are bigger than either one, so I think that is synergism. This is what I think is taking place. We have increased ammonia in both acute and chronic, inhibition of glutamate removal by the astrocyte. We're going to get glutamate accumulation, activation of NMDA receptors. I'm not going to come back to this. This is just a single signaling pathway that is affected by in a synergistic manner. Nitration of glutamine synthesis, loss of ammonia removal capacity by brain, and this cycles round to give us more ammonia. Okay? I'm not going to go into the evidence for this pathway. I don't really have time. What the manganese does is move in here. So manganese just, it's like putting your foot on the gas pedal, if you like. The whole thing speeds up synergistically. Can we ever use this clinically? I don't think we're in a position to talk about that at the moment. Example 2, ammonia and manganese again. Both activate this translocator protein in neuroglia, astrocytes and neurons. I'm going to speed up a little. Let's have a look at it. Here's translocator protein. And when activated, translocator protein switches on this neurosteroid pathway to give us allopregnanolone. I think I mentioned it in my first talk. Here's the data slide. We can see here that the exposure to ammonia gives us this increase. Exposure to manganese gives us this decrease. Gives us this increase equally of binding to this site, the bound over free, or the Bmax in this case. These were separate experiments. We never put it together. So yes, they both act in the same way, but it's not synergistic. Not obviously synergistic. We can't say it is or it isn't. So both of these compounds will stimulate translocated protein and switch this pathway on. This will effectively magnify the GABA signal. We'll get increased GABAergic tone, and I do believe that this is one of the most plausible explanations for increased GABAergic tone in HE. And it may involve synergism. It certainly could involve ammonia and manganese, at least in the basal ganglia structures. Remember, there's no measurable amount of manganese elsewhere. Here's a third example. The allopregnanolone I just mentioned to you can also act synergistically or in common with ammonia, since both are increased in the brain in chronic liver failure. Here's the allopregnanolone data that I've showed you several times, increased in HE and chronic liver failure, and they increase the binding of translocated protein binding ligand, which is flumazenil, to human brain membranes. This is an interesting story, which I think tells us a little more about how things can act synergistically. Here's the RO15 binding that was done in this, and here's the ammonia concentrations. You can see this dose-related increase in binding with 1 micromolar of allopregnanolone and different concentrations of ammonia. You can see that there's a magnifying effect. And this explains something that had troubled us a little some time ago. I'm just going to remind you from this slide that the GABA receptor complex contains modulatory sites for benzodiazepines and neurosteroids. So when those compounds are present, we get a bigger GABA signal. This was work, I believe, of Rajiv's. He showed that benzodiazepine receptors using PET in human hepatic encephalopathy were increased in patients with overt chronic hepatic encephalopathy, shown here. Okay? Increased binding sites for this ligand, which is flumazenil, flumazenil, in patients with hepatic encephalopathy. Now, we had already done studies like this before PET. We hadn't been able to find this. We'd done it with the old in vitro technique. And here you can see flumazenil binding in controls versus patients, shown here. Actually, the scattered plot is superimposable. They're so identical. There's absolutely no increase. So how can you explain that? We take a piece of brain out, we look at the receptor sites, nothing. Rajiv looks at the brain in the living patient, there's an increase of benzodiazepine receptor binding sites. How can you explain that? Here's the increase of allopregnanolone, of RO15 binding sites by allopregnanolone caused by ammonia. Now what has happened in the studies we did, we have washed out the allopregnanolone. Rajiv has done the studies. The allopregnanolone is there in situ because he's done it in the living patient. Consequently, we have not seen these increased sites. Rajiv has. What it means is actually the sites are not increased. It's the allopregnanolone in the living tissue that has made them apparently look increased by a kind of synergism. Sorry, I said that a bit clumsily, didn't I? Getting old or getting tired or something, getting jet lagged. So that was what that was about. And it's coming back to Andy. Let's talk now a little bit about, I guess, what has become an area that is of more and more interest to more and more people, and that is the whole issue of inflammation and infection. We know that we'll start out with a synopsis of the study that Andy wrote the review on. And it was that the systemic inflammatory response syndrome to infection modulates the neuropsychological effects of ammonia, the Debbie and Rajiv paper, and that there were synergistic effects of ammonia and cytokines on expression of INOS and HO1 in cultured astrocytes, some work that we did. These are two pieces of data that I know of, and there are surely others, and I've heard of others at this meeting. This is the one that I'm going to show you that we did. Again, trying to, I don't know if you've thought back to the work you did and whether you think it's synergistic or not, but you can give that some thought. I haven't done that. Here's the data we did. It's done in cultured astrocytes. These are the controls. These are gene expression for INOS and HO1 with ammonia, with IL-1 beta, and with the two together. And here's the same thing for HO1. Ammonia, IL-1, and the two together. They just make it into synergism. Just make it by the skin of their teeth. I mean, I wouldn't want to go out and draw my money out of the bank and put it on trying to repeat that or to design any kind of a clinical correlate of that. But it's there. It just squeaks in there. And so I think we are starting to see patterns emerge in terms of what this synergism between infection and ammonia is. Remember that infection and hepatocellular necrosis are both sources of pro-inflammatory cytokines. These get into the brain. They affect endothelial cells, astrocytes, and microglia. And the microglia themselves will feed back and cause more pro-inflammatory cytokines to be produced. Astrocytes swell as a result of cytokines. Endothelial cells activate, increase cerebral blood flow, edema, astrocyte neuron trafficking, encephalopathy. This is really the notion that has started to emerge. I don't want to go into that any further. Can we learn anything in terms of therapeutics? And I think it is time that we started to think about cocktails, to think about hepatic encephalopathy as being a neurological disorder, think in terms of the multifactorial basis of what it is, what it represents, and thinking about ways in which we might try multiple hits, pharmacological hits, to try to manage it. Because we know that reduction of blood-borne ammonia is only partially effective. Why would it be totally effective? We know it isn't the only issue. It's not the only game in town, by any means. The use of anti-inflammatory drugs, antioxidants, neurosteroid antagonists, NMDA receptor antagonists have been suggested. They've worked in experimental animals on many occasions. They show promise, but there's been no translational research to speak of. And I can tell you now that these compounds exist, they're on the market, they're used in similar neurological conditions. I think it's time we moved on and tried to make use of this synergistic notion, a multifactorial notion of what it is that's causing hepatic encephalopathy. Mild hypothermia actually, to some extent, does it on its own. It's effective in acute liver failure management, and it reduces brain ammonia uptake. Rajiv showed that in patients. Reduces oxidative nitrosative stress. Reduces brain inflammation. So it's a multifactorial. We're lucky that hypothermia does so much. And Javier quite clearly explained this to us in his talk. Synergism in acute liver failure. Both ammonia and pro-inflammatory cytokines are increased in brain acute liver failure, and both independently lead to oxidative nitrosative stress. Mild hypothermia is effective in reducing both. So here we, again, to put it back onto this one signaling pathway, it's not the only one that's effective. I'm not suggesting this is the only mechanism to explain hepatic encephalopathy. It is a signaling pathway that is turned on. And cytokines, through microglial activation, we know causes nitrosative stress. We get more nitration of glutamine synthesis, potentially, and even more hyperammonemia. That's a way in which cytokines can actually lead to increased brain ammonia. and we can go through this same rigmarole here with manganese and the cytokines. Just to give you a pictorial view of what I was referring to when I chose a title of Pathogenesis of Hepatic Encephalopathy, Synergism Unlimited, because it probably is unlimited. There are probably cycles like this. I just pulled this out of work that we'd done, and you could multiply this by 10 or a dozen just in terms of the many, many, many factors that have been characterized, that have been shown to be implicated. And in each one of these steps, we have a possibility of pharmacological intervention in each one of these. And I think we look forward to the day when we do start to do that. Andy said it better than I could. Andy always said everything better than we could, didn't he? He always had the right words and the right way to say it. Over the years, he said in his editorial, several theories have been proposed to explain the mechanism responsible for HE, and the search has been reinvigorated by a new perspective on synergism. Just one message out of the 100,000 messages that Andy Blei left us with, and we are most grateful to him, and always will be. Thanks, Andy. This was in Newcastle. We miss you. We miss Andy. This was Walking on Hadrian's Wall. We miss Juan, and we'll talk about that at some other time. This was Walking on Hadrian's Wall. We're all sort of bundled up. We found a little bit of sunshine there. It was probably in mid-July or August. We had a great meeting in Newcastle at the time. I'm very grateful to ISHEN again for giving me this honor, to honor Andy in this way. And I'm grateful to these people who, including Andy. And let me mention Gilles Pommier, whom we lost also long before his time, also just a few months ago. And to the many people of this organization who've been so generous to me, to Andy, and Andy to us over the years. Thank you very much for your attention.